Hello everybody, we're gonna try to go over chapter 18. This is a chapter that deals with the genetic material, genetic makeup of viruses and bacteria, and we just need to know some generalities about them. Please notice the beautiful photograph of a bacteria with bacteriophages attached to it. Very nice. And then some leaves there showing some virus damage. So yes, plants do get infected by viruses. So let's start with a nice view of the size differences, and this is important because we can only see under the regular light microscope, we can only see eukaryotic cells and prokaryotic cells. Viruses are very, very small and we are not going to be able to see them. The only way we can see the viruses is using the electron microscope. Now, for viruses, that's the first thing that we're going to do. Viruses, basically, their structure is fairly simple. They are not living organisms. They are just infectious particles. Basically, the only time that they technically kind of come alive is when they are inside a host cell. So viruses really are made out of two main things, most of them. Is a nucleic acid that can be DNA or RNA, and surrounding the nucleic acid as a source of protection is what we call a capsid. A capsid that is basically a congregation of proteins, a lot of proteins together making like a protective barrier around the nucleic acid. So that's the basic structure. Here you have electron micrographs, and here you have an idealized picture showing the structure. So notice this is a mosaic tobacco virus, one of the very first ones that was studied, infects tobacco plants, of course. And this one has RNA. Then we have, this is an adenovirus. Adenoviruses are usually the ones that cause upper respiratory infections. Notice that it has DNA and is surrounded by the proteins. Then this one here is the flu virus, the influenza. This is a slightly different one because you have the RNA, the nucleic acid is RNA. The RNA is surrounded by the protein protective layer, the capsid, but then it also has an envelope, a membranous envelope that is made out of a piece of membrane from the host cell, the kind of the steel membranes from the host. And we'll look at this in more detail in a minute. Notice these glycoproteins that are attached on the outside of the membrane. These proteins are made by the virus. We'll see that. So this is influenza. It's also the same structure for the HIV virus. So important one. And then over here you have the one that you're familiar with, the bacteriophages that specialize in attacking bacteria. And the DNA is inside. And then all this other structure is just protein, part of the capsid, and they are very complex because these ones, they don't penetrate the cell, they just inject the DNA into the cell. These other ones actually go into the cell, so they have a different structure. So just to give you some idea of those types, in general, very general principle, general cycle. You have a virus, in this case we have a DNA virus with the capsid, this protein surrounding it. The virus is going to penetrate a cell. As soon as it goes in, actually, the capsid is broken apart. The individual proteins that make the capsid just disappear, not disappear, separate, and the DNA or RNA, whatever nucleic acid is released. In this case, we're dealing with DNA. So what's going to happen to this DNA inside? Well, first, some of it is going to be copied, DNA replication, to make more copies of it, all using the host machinery. So they use all the host enzymes. All the enzymes that we use for DNA replication are basically used by the virus. They don't carry anything. They use whatever is in the cell. So they use your nucleotides, your helicase, topoisomerases, DNA polymerase 1, DNA polymerase 3, all of them. Oh, and your ATP, everything. They use everything that you have in your cell to make more copies of themselves. So this DNA, some of it is replicated, DNA replication using all your basic products inside the cell, while some other part of it is transcribed into messenger RNA and then translated into those capsid proteins. So you're doing DNA replication and also transcription and translation. Good? So all this using the ribosomes, using everything that we have inside our cells. So basically the viruses come in and take advantage of everything that we have, and then they're going to leave. Just like those guests that come to our houses during the holidays, take advantage of everything and then they say goodbye. So here you have copies of DNA, copies of the protein using your ribosomes, and then all these actually kind of self-assemble at the end. The capsid congregates, think of like little magnets coming together. The proteins come, and inside is the DNA, and then leave the cell, and that's the end of it. Basically, one comes in, many go out, and that's the end of it. Right? An important thing about viruses is that they are very host-specific and cell-specific. Host-specific, they infect usually just one or a few species that are closely related, species of organisms. But then even within one particular host, every virus is very specific about the type of cell that they are going to infect. So for example, adenoviruses infect the upper respiratory and only attach those cells that they have specific markers on the surface where they can attach. HIV virus, you might get HIV all over your body, but the virus is only going to infect when they find the markers in your white blood cells. Same thing if you get a flu virus, for example the stomach flu, you might get it everywhere in your body, but the only cells that the stomach flu virus is going to attack is the cells of the epithelium of your stomach. So very specific because they recognize specific markers. Remember, every cell has, hello, my name is. They're going to recognize those specific markers there. Now, in general, so this is the overall pattern that viruses have. In general, most viruses have two types of reproduction that we call the lytic cycle, short word, short life. And the other one is a lysogenic cycle that we'll look also. Let's start with the lytic cycle. Then we'll compare both. Lytic cycle. This cycle usually ends with the lysis or death of the host cell. That's why short word, short life for the host. So using the model of the bacteriophage, the virus attaches to the bacteria cells, the virus injects its DNA, and then uses the bacterial machinery to make copies of its own DNA and to make copies of all the proteins, basically to do transcription and translation. When all the elements are inside the bacterial cell, these elements basically self-assemble, and since the machinery keeps on making and making more viruses, the cell just cannot hold them anymore and poof, blows up. And that's the end of the cell, and all the bacteriophages get released and they are free to go and infect another cell. So this is the lytic cycle. Basically, they die. Now, in contrast to the lytic cycle, you have what is called the lysogenic cycle. Here, instead of killing the cell, they do something that I find very creepy, really. And the best way to do it is looking at a picture. Basically, same process, you have a bacteriophage that infects a host cell, but this time, the DNA of the virus, instead of doing replication and transcription and translation, what happens is the DNA of the virus is going to join the DNA of the host cell, the DNA of the bacteria in this case, and becomes part of the bacterial DNA. Now, since DNA is all the same, A, T, C, Gs, the structure is all the same, the cell never really knows that it has DNA from somebody else. It treats it just like as DNA. So what happens every time this cell divides? Well, before division, it copies its DNA, and besides copying its own DNA, it's also making a copy of the virus DNA. So when the cell divides, each cell has a copy of the virus and its own DNA. So as long as they keep on dividing, you keep on passing this viral DNA from one cell to another. But the good thing, you're not dead. The bad thing, you have something inside you that doesn't belong to you and you just keep on passing it. Basically, that's life. That's what you're always trying to do. Pass on your DNA to future generations. However you do it, doesn't matter. Very clever method here of doing it. So this is the lysogenic. You just can keep on hiding in the DNA of the host and never do anything. However, sometimes, triggered by environmental conditions, the virus might switch out of the lysogenic cycle and decide, okay, that's enough. And basically, instead of being quiet there, it starts replicating just itself and it starts doing transcription and translation of its own capsid proteins, produces all the stuff that they need, capsid plus copies of the DNA, they assemble and poof, they die. So you can switch from lysogenic to lytic cycle. Something similar to this that we have in ourselves is the herpes virus. It doesn't work quite the same, but it's very similar, good enough to understand the process. When you get those sores in your mouth, or if you get the genital herpes, when you have a breakout, that means that your virus is doing the lytic cycle. The breakout is because your cells are broken out and breaking out, and you have viruses being released. Then all of a sudden, you seem cured. Nothing happens. Oh, you got rid of the virus. Not really. Because the virus can hide inside your cells and stays there for a long time. And during that time that the virus is hiding, you don't have any symptoms of anything and you seem to be cured. So when it's hiding, you are going through kind of the lysogenic cycle. Now, usually triggered due to some environmental factors or stress, or you eat something that is very spicy, that doesn't sit well with you. We are not sure exactly what, but that is a trigger sometimes for the viruses to come out of the lysogenic cycle and become lytic. So, you don't have anything, and then all of a sudden, without explanation, you break out. Well, your virus that was just sitting there, all of a sudden became lytic. So they can switch back and forth. Some of them hide, some of them don't. So different viruses do different things, but these are just generalities that apply to many of the viruses. Interesting life. Very interesting life indeed. Alright, so let's skip this one and go directly to RNA, because the RNA viruses, in particular a group that we call the retroviruses, are very interesting and very important for us for two reasons. One is HIV is a type of retrovirus, and also out of these viruses we are going to get a particular enzyme that is going to be very important for all our biotechnology industry. That enzyme is called reverse transcriptase. So let's look at what a retrovirus is like. First, it's an RNA virus. And you can see here, if we start on the inside, you have two RNAs. That's their nucleic material, RNAs, and they have two molecules. They also have an enzyme, and they carry their own copy of the enzyme. And the enzyme is reverse transcriptase. Remember the name, reverse transcriptase. Surrounding that, of course, you have the capsid made out of proteins. And then surrounding this, you have an envelope, the envelope that is mostly made out of phospholipids that came from the host cell, but also has these major glycoproteins that were coded for by the virus itself. Now, if you recall, the process of the central dogma that we call goes from, the central dogma goes DNA to RNA, and then of course to protein. Now notice this name, reverse transcriptase. Reverse transcriptase, as the name implies, what it does allows you to go from RNA to DNA. It goes backwards. So this is kind of an exception to the central dogma. So this reverse transcriptase is the enzyme that allows you to go backwards. And they carry their own enzymes with them. So how is this going to work? Alright, let's look at how HIV does it, and this is in very general terms. You have a beautiful diagram like this in your textbook that you can see with more detail and has little boxes that explain it, so please use it. So if we start with HIV, actually HIV, since it has that membranous envelope around itself on the outside there, what it does is that envelope fuses with the membrane there, and basically it's kind of like doing endocytosis, kind of. Basically the membranes fuse and release the capsid. Notice the capsid, that is going to be released, and then as soon as the capsid is in the cytoplasm, the capsid disintegrates, all the proteins disintegrate, and it releases the two molecules of RNA and the two molecules of the two enzymes. What happens next? The enzyme, reverse transcriptase, starts with the RNA of the virus and makes a copy of DNA, makes a transcript and goes back to DNA. And then this goes over again through reverse transcriptase and makes the second strand of DNA. So it does it 2 times. You can look at the details in the book, and basically you end up with a double-stranded piece of DNA. This piece of DNA now is going to go into the nucleus and it's going to join the host DNA. It's going to be inserted into the host DNA. Very nice. What happens when it's inside the host DNA? Well, it's going to do transcription, but some of the transcription is going to be transcribed into messenger RNA that is going to code for 2 specific proteins. The proteins, the glycoproteins that are going to form part of the envelope outside, and some of the mRNA is also going to code for the protein code, and some RNA is also going to code for reverse transcriptase. So it makes all these individual components. On the other hand, some of this DNA is being transcribed, the whole thing, into RNA as the nucleic acid. So transcription, regardless, doesn't matter if you are making messenger RNA or just a copy, a plain good old RNA, is transcription. So those 2 things are made, the nucleic acid plus all the proteins, they assemble into that capsid, and then the capsid leaves the cell, and as it leaves, grabs a piece of that membrane with the virus proteins, and there you have it, a brand new virus. Now, why is this important and why they are called retroviruses? Well, they are called retroviruses because they go from RNA to DNA, they go backwards. Now, when they do that, the DNA that they make is not just called DNA, it's called cDNA. cDNA for complementary DNA. Complementary DNA. Because it's complementary to the RNA. What's the other thing that is significant about this? The other part is that this section here, where you are using reverse transcriptase, every time that RNA is copied into DNA, it's copied, but there is nothing that can proofread that sequence. You know, in the nucleus we have all these proofreading enzymes like DNA polymerase 1 that checks to make sure that you didn't make any mistakes during the copying process. Well, here you don't have that. So this is a section of the cycle of the virus that is very prone to mutations because they make a lot of mistakes. What happens when you make a mutation here, when you make a mistake? That means that the proteins that you might make are going to be slightly different. And that means that the glycoproteins outside are going to be slightly different every time, as well as some of the other components of the virus. Now, what's the importance of this? The importance of this is that when we make vaccines, when we use or have a vaccine basically is a piece of the pathogen that we insert in our body to teach our body what to recognize, to teach our immune system to recognize something that is foreign. That's what a vaccine is. We inject either dead viruses or pieces of bacteria. That's what we are getting. Well, we cannot do that with these guys because they change so fast because of this high mutation rate here that I can teach your body what this HIV looks like today, but in a couple of months it's going to be different. Your body is not going to be able to recognize it because of the high mutation rate. That's the significance. Which other viruses do this? Which other viruses are retrovirus? Well, the flu virus is also a retrovirus. If you think about it, how often do you need to get flu shots? Once a year. Because every year we get a vaccine based on the most common strands of the previous year. So they pick those, they make the vaccine, they teach our body in the hope that we get infected or we get exposed to one of those and be protected. So they have to do it every year because they change that often. That's significant. And reverse transcriptase, the other important thing, does allow us to go back from RNA to DNA. You have a beautiful picture of HIV virus going into a cell. You can see it penetrating the cell, and in this side you can see the viruses coming out. You can even see the capsid inside. These are beautiful shots. So beautiful pictures. I encourage you to go to the PowerPoint and look at this video here that I marked as awesome because it's truly, truly unbelievable. It's 5 minutes. You might have to watch it twice, but it gives you everything that you need to know about how HIV works and how the drugs that we are using to stop HIV or to stop the infection really work. Very, very, very good. Please watch it if you have a minute. And finally, just to round out the virus business, remember that plants also get infected with viruses. And I just want to touch on transmission of viruses and make sure that you understand the difference between horizontal transmission and vertical transmission. Horizontal transmission in general is what I do when I sneeze and I spread all my viruses everywhere. I just give it to everybody, related or not related. Yes, that's horizontal. In the case of vertical transmission, is something that I give directly to my offspring. And that would be, for example, if I'm infected with HIV and I have a baby, and the baby is born through the vaginal canal, when they come out with all this mess out there, they pick up the virus. That would be kind of a vertical transmission because I pass it directly. But in plants, you can also have this, and humans are usually very guilty of horizontal transmission because we work on a plant and then we use the same tools to work on another plant. We are passing it along very easy. So, simple concepts, and that puts an end to the virus. Please take a minute, stop, meditate, process the information, make sure that you understand really the basics of viruses. When you're ready, pick it up again and we'll do bacteria genomes. So, bacteria. Bacteria are interesting creatures, prokaryotic cells. They have no defined nucleus, and for genetic material they have what we call the bacterial DNA, that is a circular piece of DNA, relatively big, has about maybe up to 20,000 genes, but in addition to that, in addition to the main chromosome that is one circular, bacteria also have smaller pieces of DNA, also circular, that we call plasmids. So main chromosome, but they can also have plasmids. And the beauty of these plasmids is that bacteria are pretty good at sharing these plasmids from one to another. Even if you have the environment plasmids floating around, bacteria like to pick up those plasmids. So these are pieces of DNA that carry just a few genes, maybe 1, 2, 3, 4, 5 genes, so very small compared to 10, 20,000 genes. And we are going to be using this a lot in, again, when we do biotechnology, so keep in mind about that. Before that, we talked about how DNA replication occurs in prokaryotes. Since it's a circular DNA, we don't have the problem of telomeres. You have the origin of replication as usual. You start copying it there and you go in both directions. So you still have to deal with Okazaki fragments here. And this keeps on opening, opening, opening until basically you complete the whole circle and these two pieces of DNA just separate. Because you only have one circular piece of DNA, you don't need to go through the whole process of mitosis where the chromosomes have to align neatly and separate. There's no point, you only have one. So it's a simple division, simple separation of the chromosome with the cell. And that's the basic concepts about division in prokaryotes. Now, bacteria, since they divide this way, and we know that that's a type of asexual reproduction, that means all the offspring are exactly the same. And by now we know that that's great when the conditions are perfect because we all do well, but it gives a very little buffer for when the conditions in the environment change. So, how do bacteria manage to increase their genetic diversity to be able to survive when environmental conditions change and to be able to deal with all the things that the environment throws at them? Well, they have ways to do recombination, actually, to change their DNA besides mutations. They do other things besides mutations to increase their DNA. Please remember that mutations are totally random. Some are good, great, you survive. If you get a bad mutation, basically you are not going to make it and we are not going to see. So sometimes mutations are also neutral, so it doesn't matter. So that's the problem with mutations. Good, you survive. Bad mutation, you are out of the gene pool. So bacteria are always looking for ways to increase their genetic diversity. Since they divide by simple division and they are all clones technically, they increase their diversity. And they do it using three processes. One is transformation, the second one transduction, and the third one is conjugation, and that's probably my favorite. So let's go over each one of them, because pictures are the best way to explain this anyway, as usual. So, transformation. Transformation basically is the uptake, is bacteria are up there and they are always looking for little pieces of DNA in the environment that they can pick up. So there they are, oh, piece of DNA taken, oh, DNA taken. So they are always picking up foreign pieces of DNA that they encounter. So if you have a cell like this that dies and the DNA disintegrates and is all over, cells in the same culture or in the same environment can pick up little pieces of DNA. And they can integrate that DNA to their genome. If it's a good gene, they're going to keep it and survive. Sometimes if they pick up something bad, they die and we never find out about it. So a little bit of a chance event, but this is actually how, if you remember Griffith experiments with transformation, the first time that we realized that DNA had something to do with genetic information. Remember that we had the good bacteria that became bad bacteria. Well, the good bacteria became bad bacteria because they picked up the gene to make that capsule that protected them from the immune system of the host. So this is transformation. And we're actually going to do a lab in January. We're going to transform bacteria and see what happens to them. Please encourage you to watch the little video. This is just a 1 minute little shot, but it's very useful. So transformation, that's what you have there. Next, transduction. Again, remember we're trying to see ways how bacteria can enrich or can modify their genetic material. And transduction is actually pretty interesting because the bacteria don't do anything. It's a side effect of being infected with viruses. And again, the best way to understand this is with a visual. So you have a bacteria, and in the genome of the bacteria you have A plus and B plus there. So I have identified two genes here, those two genes. This bacteria is infected by a phage, and this is part of the lytic cycle here. So the whole DNA of the bacteria breaks, the phage takes control over the cell, makes more phage, but something interesting happens. Since the virus particles assemble, just come together, sometimes when they come together, besides the virus DNA, they can pick up little pieces of bacteria DNA, just like collateral damage. They get trapped there. What happens now if this bacteriophage infects another bacteria? When it's releasing its own DNA, it's going to also release that piece of DNA that they took by mistake from the other bacteria. Now, if this DNA happens to be complementary, they are homologous, it matches a sequence, they are going to match up, actually line up together like homologous chromosomes during crossing over in eukaryotic cells. They are going to match up, and just by chance again, you might have crossing over there. If you have crossing over, basically what you're going to do is you're going to replace the existing version of the gene, A minus, by the new version of the gene that is the A plus. So the bacteria at the end ends up with an A plus and the original B that it had. Now, all this only would happen if this virus doesn't kill the bacteria cell. You know, sometimes that happens because the bacteria have ways to defend against viruses. And those ways to defend themselves are called restriction enzymes. So, if it survives, now we have a brand new bacteria cell that has a slightly different gene, and it has become slightly different. So that is transduction. And the virus in this case is called a vector, because a vector is something that transfers materials from one place to another. So the vector is transferring the DNA from one cell to another. It doesn't affect the virus, it's just the vector. Another vector that you know, for example, are ticks. Ticks are vectors for Lyme disease, for Borrelia, whatever the name of the species is. Basically, ticks are not affected by the bacteria, but if we get a tick that bites us, they transfer the bacteria to us, Borrelia, Dork's furry, whatever the name is, and we get Lyme disease. But the ticks are not, so the ticks are also vectors. Mosquitoes are also vectors for malaria. Mosquitoes do not suffer from malaria, but we do when the mosquitoes give us the protozoa. Good? So we had transformation and we've had transduction. Transduction accomplished by a virus. The last one is conjugation. Conjugation is basically the transfer of plasmids, of DNA material, mostly plasmids, between cells. And once again, a picture is the best thing. So to do the conjugation, you have two bacteria cells, and what they are going to do first is they are going to make a connection. And that connection is accomplished through an extension of the cell membrane called the, let me write it up here, pilus. That's singular, and plural is pili. Because sometimes you have many of them connecting with each other. Here I only have two bacteria. You might have 3 or 4 and they are all connecting with each other. Quite a party. Now, what's going to happen here? What's going to happen here is you have a bacteria that has the main bacterial chromosome, that is never touched, but then you have a plasmid. And this plasmid, what the bacteria does, they connect, through the pilus, that is also called a mating bridge. It duplicates its plasmid, copies it, and then through this bridge, it sends a copy of the plasmid to the other bacteria cell. So at the end of the process, this one kept, of course, its original plasmid and gave a copy of the plasmid to the other one. So now they both have that plasmid. What is so good about this? Well, whatever information was here, now there are more cells that have that information. And obviously, if this cell was able to survive with this, it's a good gene and it's passing it around to the other ones. Now, in order to do this transfer, the bacteria have to have a gene called the F+ gene. F stands for fertilization. So if they have the F gene that is going to allow them to do this, what they are going to do, they are able to transfer. Notice that the transfer is only one way. From a bacteria that is F+, is going to be able to transfer to another bacteria. So the F+ is always the donor cell. Shoo, send it. Now, once the F- or whatever that didn't have this kind of gene, the F+ gene, gets it, guess what? Now they are F+ and unable to keep on passing this to other ones. So once you get it, you have it, and you become part of the F+ group of cells that can keep perpetuating this plasmid all over. This is really cool because actually a lot of the we have a lot of problems with bacteria and antibiotic resistance. You know, you try to kill them with antibiotics and they don't die. Usually they don't die because they have a gene that breaks the antibiotic and renders it ineffective. Now those genes, a lot of the genes that help the bacteria survive antibiotics, a lot of those genes are carried in plasmids. So picture yourself, you have a strep throat infection with the Streptococcus pyogenes, you have all these bacteria here, and imagine that you have one bacteria that is resistant. Now what happens when that bacteria starts giving its plasmid to all the other bacteria in your strep throat? Well, all the other bacteria are going to get this F+ plasmid that makes them resistant to the antibiotic. You take your antibiotic and you're still sick. So what has to happen? Then you have to take a different type of antibiotic that they are not resistant to get rid of your strep throat infection. This is the most common way that bacteria are passing some of these resistant genes from one to another, and that's one of the problems that we are facing, that is hard to stop it because of this. So bacteria like to share their genes, and they're very promiscuous. Sometimes it's 2, 3, 4. This is conjugation. The way I remember conjugation always is I think of conjugal visits. If you know anything about this, people in prison, depending on the prison, if they are married, they are allowed to have their spouse to visit them, and they get a private room and the whole thing. That's called conjugal visit. So I think of conjugal visits because they are actually here conjugating, doing their thing, sharing their genes, and making something new. Alright, that's how I remember it. You try to come up with your own method to remember transformation, just picking up DNA from the environment. Transduction, that's when the virus works as a vector carrying DNA. Or conjugation, this is kind of the equivalent of having sex, really, for bacteria. Remember, sex, basically what it does is mixes your genes a little bit. Here, that's why it's called a mating bridge, because they are mixing their genes, although kind of unilaterally. Good? So that's what we have to do with viruses and bacteria. Please take a minute and watch all the little videos in this series, and they are all like a minute long, except for the one for viruses. I hope this helps.